So thanks a lot to the organizers as well. I'm really sad that we can't all be there in person in Freiburg. This would have been a really great community to interact with in person. So we make the best of it online. And yeah, you can follow us on Twitter. Here's the handle at Nash_lab if you're interested in this research. And I'll talk about engineering biophysical properties of therapeutic proteins. We don't do only rational design. We also do somewhat irrational experiments as well, but we'll get into that. I need to acknowledge definitely a postdoc, Hai-Pei Liu, and PhD student from my lab who just graduated, Xiaowei, who were really the key players on the projects that I will present today. And Xiaowei has now moved on to bigger and better things as a postdoc in Nienke Decker's lab at Delft, and he will be missed in the group, and you'll see some of the really great work that he was able to complete during his PhD. So just a brief introduction and overview of my lab. We have two main themes that we work on. One is molecular mechanics. So here, as you're all familiar with, this is the AFM. We do single molecule force spectroscopy on engineered proteins, and we're interested in how force can change the function of proteins, how it can modulate activities like binding activities. We're interested in intermediate folding states of proteins, influence of post-translational modifications, and so on. And we do this all using sort of the current best state-of-the-art in surface chemistry and site-specific linkages and protein engineering. The second part of my lab is focused on engineering proteins using polymers and hydrogels. So we can use different types of hydrogel materials in protein engineering projects to accelerate things like directed evolution based on localized formation of hydrogels around single cells, and also working on systems that are based on elastin-like polypeptides for blood clotting. And there's a lot of interchange and overlap between these two topical areas, but this is kind of how we see our different programs that I have in the lab. So changing gears from the prior talks where we learned about, really a physical chemistry and tribology perspective, now we're talking about biological activity of mechanics in protein systems. So there's plenty of examples to point to. So for example, this work from Sergei Garcia-Magna's lab in University College London showed that the transport rate of transcription factors in and out of the nucleus could be controlled by fusing these transcription factors to known domains with different mechanical properties. So the mechanical shearing or the resistance to mechanical unfolding as it translocates nuclear pore complex is known to somehow control that translocation rate into the cell nucleus. Also, this is from the Gaub and Bernardi labs. This was the work of Lucas Miles, who's a speaker at this conference, also later on the program, showing that this pathogenic adhesin protein, SDRG, is, I believe, sort of this class of proteins represents now the most mechanostable non-covalent interactions that we know of. These can withstand about 2 nanonewtons of tension before they break. So it's really incredible forces that can be generated or resisted by conformational changes in proteins that provide mechanical stabilization. Another example I would point to is Wesley Wong's group looking at tip-link proteins in auditory perception. So the way we perceive sounds and vibrations from the environment in our inner ear is related to the mechanical properties of these tip-link proteins. So it's clear that for natural biological systems, like in the human body or in pathogenic bacteria and so on, that mechanical properties can play a very important role. And so now the question is, well, what about synthetic and engineered systems? So this is really one of my passions, is sort of artificial synthetic biology with a focus on biophysical properties. How could we design systems that could take advantage of some force and function of proteins in order to develop new types of medical therapies? And the context for this is the rise in biological therapeutics, drug approvals that are based on biologic drugs. You can see that's really going up. Of course, the huge success of 2020, which is also a biological therapeutic, of course, is the coronavirus vaccines. So this is really just, a triumph of modern molecular engineering and biotechnology that we have these effective and safe vaccines. And this trend is only expected to continue in the coming years. So what do we expect in terms of developments of biological therapeutics? Well, we're moving from these small molecule systems where we have well-defined structures, and we're moving towards more complex systems, more atoms, larger molecular weights, like this antibody drug conjugate, and then even more complex systems like enzyme precursors, so zymogen precursors. So this is a transcription, or excuse me, not a transcription factor, it's a coagulation factor, factor 8. It was approved in 2019. It's pegylated at a single side chain. It's not active as a stored compound. When it enters the bloodstream, it can become activated by endogenous proteases in the blood. So we're developing more advanced types of biotherapeutics. And what's interesting is that now, as we go away from defined structures, our therapeutics are going to sort of exist on this continuum of structure and conformation, and the aspect of biophysical stability becomes more and more important. So what does that mean, biophysical stability? Well, it refers to sort of a broad range of properties of therapeutics, and one of the concepts is that of developability. So how easy is it to develop a candidate molecule all the way through, get it through clinical trials and approved? One of the things that companies are doing is they're applying a range of biophysical assays, and these are things like colloidal stability assays, assays to measure nonspecific adhesion to common protein antigens in the bloodstream, many different ones. So in this paper that I'm citing here, this came out of Adimab, which is a spinoff from Dane Wittrop's lab at MIT, and they did 12 different biophysical assays, like some of the ones I mentioned, and they tested 137 different monoclonal antibodies that had been entering into the clinical trial pipeline. And some of these monoclonals had failed at different phases, like phase 2 or phase 3 clinical trials, and some of them had made it all the way to approval. And when they tested these, they found that for candidate molecules that scored in the bottom 10% of that cohort of the MABs, they would give that what was called a red flag. And you were much more likely to reach approval if you had 0 red flags in these biophysical assays. So this just tells you that the biophysical stability of these molecules is somehow predictive of their ability to make it through a clinical trial pipeline. But of course, all these assays are performed at equilibrium. That's a really important aspect to mention. And so the question is, how about non-equilibrium mechanical stability? So non-equilibrium properties of biologics are currently neglected in biopharma development pipeline, but if we could accelerate this or get better insight into biophysical stability parameters at an early stage in development, maybe this could be an advantage for this type of pharmaceutical development. So with that, I will get on to just sort of two aspects of my talk. I'll talk about some of the methods development that we're doing for single molecule AFM stretching and pulling and unfolding of proteins. And then I'll tell you one short story about optimizing the anchor points to achieve higher mechanostability in a cancer therapeutic. So my lab is sort of using, the best current methodology for AFM, and this has been contributed to also by various people and groups, so I don't claim ownership over any of these methods exclusively. We're also happy to share methods and especially plasmids to the community as well. But the way we do this now is with just a force robot system from JPK, and then we have these, of course, cantilever tips, which everybody in this audience is already familiar with, and then we have sort of back-end processing software to analyze curves. So we collect tens of thousands of curves, and then we filter them for valid single molecule interactions. So a lot of the interactions that we observe in the experiments are due to non-specific interactions, or they can pull multiple molecules in parallel. And we're interested in capturing the behavior of single receptor ligand molecules or single proteins unfolding under force. And we do that by collecting large amounts of data and then filtering and actually throwing out the majority of the data. But based on internal control elements that we build into these systems, we can validate single molecule interactions and then analyze the behavior. So one of the methods that was developed early on and that came from other polyprotein literature was the idea of using fingerprint domains. And so in this experiment, for example, we're interested in the mechanical stability of an antigen, in this case, a fluorescent protein with a single domain antibody called the VHH domain. And in this particular paper, we were studying the influence of disulfide bond formation on the VHH M-cherry interaction. And we built this with these fingerprint domains. So these are fusion domains to the antigen and the antibody. And when we ramp the force up in the single molecule AFM, we see these characteristic unfolding patterns that can be mapped into contour length space using elasticity models. And this allows us to assign increments and unfolding events that we see in the trace to different molecules in this polyprotein, and then finally quantify the rupture force distribution. This just gives you an impression of the type of methodology. Coming from inspiration from Tom Perkins' lab, we've started modifying the cantilevers to give us better time resolution and better stability. So this is called the Warhammer design, and Tom Perkins' lab has been really helpful in getting this up and running, but these are actually relatively straightforward to fabricate. If you have access to a focused ion beam in a scanning electron microscope, then you can take commercial cantilevers and improve their properties, and that can help with the measurement, as you see here, the lower noise and so on. In terms of bioconjugation chemistry, we've done a lot of work there, sort of implementing existing protein ligase chemistry or SpyTag SpyCatcher, and sort of utilizing this in building up controlled systems, molecular systems, where we really can define every atom in the system. And so this is some of the work on that, like these spontaneous isopeptide bond formation between SpyTag and SpyCatcher from Mark Howarth's lab. This is extremely useful and gives us a modular approach towards building up surface chemistry. Sortase also is a protein ligase that can be used to oligomerize domains into artificial polyproteins. And then, as I'll show in the second part of the talk, then this bioorthogonal click chemistry can also be used. This is a slide here on the left. This is showing how we can build up systems that have force responses in really different ranges. You can build up polyproteins that have low, medium, and high force unfolding events. And what's really nice about working with very high force interactions is that when you transform that into contour length space, you can start to pinpoint distance lengths very precisely down below about 7 nanometers is really doable with our current systems, and that's with the commercial cantilevers. We've also developed some analysis algorithms that can correct for certain types of artifacts that occur in these experiments. For example, if you unfold a marker domain using a receptor ligand to apply force, then what happens is you actually bias the rupture force distribution of these complexes, or excuse me, of these fingerprint domains. That's just based on the intrinsic stability of the receptor ligand. In a certain fraction of the traces, the interaction that you're using to pull on a molecule will break before the domain itself unfolds. And this leads to a statistical biasing effect in the unfolding force distribution. So if you do an experiment like this, it's actually not possible to directly observe the true unfolding force distribution for this fingerprint domain. We've developed a correction algorithm that accounts for the fraction of traces that have this fingerprint, and you can use these types of correction algorithms to extract true unbinding energy landscape parameters for that fingerprint domain. We use elastin-like polypeptides as linkers. These are intrinsically disordered proteins, so we can eliminate some of the artifacts that come from polydispersity of synthetic polymer systems and that are associated with some sort of mechanical isomerization in polyethylene glycol, for example. This can be overcome by going to intrinsically disordered proteins as linkers. And this is sort of our best current state of the art in surface chemistry, where we can use a very modular platform, and our SpyCatcher DDFLN4 fingerprint domain construct is sort of universal for all the AFM. Then you have this SpyTag and then the protein of interest that you can express as a SpyTag fusion, and then you can put this into this modular surface chemistry and then analyze. I showed you contour length transformations. We can also use machine learning and pattern recognition to sort and classify traces into different unfolding patterns. And this works like as sort of your Faces app in your iPhone. If you know this, you can train a deep neural network. You can tell it what certain repetitive unfolding patterns look like, and then this deep neural network will process the rest of the curves and show you similar curves. And then you can project this into lower dimensional space and start to segregate your data based on different unfolding pathways using neural networks. Again, with no input of physical properties or any kind of elasticity models. This is just based on actually image analysis. Let me get to the second part of the talk, and I'll try to speed up a little bit and maybe we catch up on the time. This is available as a preprint right now on bioRxives, but here we're interested in this concept of mechanical anisotropy in proteins. So here the concept is that force is not only a scalar quantity, it's in fact a vector quantity. It means it has an associated direction. So depending on how you pull on molecules, they can have very different types of mechanical responses. This was shown for polyubiquitin chains, for example. This is work from Matthias Rief's lab. So if you link GFP and you assemble it into oligomeric polyproteins through disulfides, then you can stretch this GFP through different axes, and you see that there's very different force responses, force on the x-axis. We've shown some of this work with mechanostable adhesion proteins, and of course, shear and zip geometries of DNA are a classic example as well. And this works exactly like Velcro. You can shear apart, you can unzip the Velcro, or the DNA in this case, at low forces over long distances, or you can dissipate the binding energy over shorter distances, but at, of course, higher forces. So what about protein-protein interactions? So there's been other work in this. I should really point to Stefan Sedlak's work from the Gaub chair at LMU, looking at streptavidin from the anchor point perspective. But here we're actually doing a slightly different experiment where we want to scan the anchor point across the entire backbone of the protein sequence. So in this case, we took a cancer therapeutic protein called CTLA4 anticalin. So this binds to the immune checkpoint inhibitor called CTLA4. And we're just asking the question, okay, what anchor point is the best to pull on this anti-CTLA4, this anticalin molecule? Where would I best apply force if I want to maximize the dissociation forces between this red molecule and the antigen? And so how are we able to build up this chemistry? How can we actually pick up molecules from individual internal residue positions? We can't just fuse it to different peptide ligase domains, for example, because that wouldn't work. So we needed to come up with a bit of a clever way so we can use bioorthogonal click chemistry. So we use non-natural amino acid incorporation through a technique called amber codon suppression. So it allows us to introduce bioorthogonal click groups into a protein at any position we want. And this reduces the protein expression levels a lot, but if you can get a cooperating system, you can generate enough quantities of these compounds, and then you can synthesize this peptide with a DBCO group. And this peptide is very special. It forms a very mechanostable bond with its target called SDRG. This shows mass spec and SDS page data showing that we can produce these compounds and purify them.
You have 3 minutes left.
Thanks. And the trick to this measurement is to use this as a freely diffusing system. So we have the antigen tethered to the surface through our fingerprint chemistry, and then we can add these molecules of anticalin with this sort of T-junction within the protein, this Frankenstein type protein where you have a peptide sticking off an internal residue number of that protein. You can pick up the protein at that position with SDRG and then collect large amounts of rupture force data. And since this complex is a mechanostable bond, whereas this one is much weaker, then we're probing the ruptures of these complexes. This guy rapidly exchanges in solution, and so it frees your tip, and so you can produce these measurements. I'll get to the business. So all the traces essentially look the same because they all contain the exact same fingerprint set. But what's different is depending on the anchor point residue, we have an increase or a decrease in the rupture force of that protein-protein interaction. And if we plotted the mean rupture force as a function of the anchor point residue along the anticalin backbone, what was quite interesting is that it wasn't sort of random or randomly distributed, but we really see a sort of a structure. It seems like in the middle of this molecule, if you pull on it from the internal sequence positions, you're able to achieve high rupture forces, where if you peel it away from the termini of this molecule of CTLA4, it really is sort of more like an unzipping transition. And then we see this in the energy landscape parameters, and we see this also in coarse-grained molecular dynamics simulations that we are collaborating with Adolfo Poma in Poland on. And so this type of data, I think, is kind of new to the field, where we can probe the rupture force distribution as a function of pulling point along the backbone. And you see that you could be pulling on this molecule from much better positions other than the N or the C terminus. So this shows you that there's actually mechanical affinity maturation that you can achieve for these types of binding proteins. You can do this across a range of loading rates and extract energy landscape parameters, and you can depict the energy landscape in different types of ways, where the directions along this landscape represent pulling geometries, or you can represent it in this way. You can see that there's huge differences in that intrinsic off rate at given force. For example, 100 piconewtons, you can see 2 to 3 orders of magnitude changes in that intrinsic K off parameter depending on the pulling point. So I think that's quite an interesting finding, and we were excited about that. And with that, I'll summarize and conclude. We know proteins are soft molecules. They deform anisotropically depending on a very complex network of intramolecular interaction potentials, and also intermolecular, right, between two binding proteins. And so we're developing new methods for site-specific pickup of these molecules, and we see large differences in the stability of the complex, the mechanostability, depending on how we pick up these molecules and stretch them. And we think this has potential applications in drug delivery under shear stress, and we are pursuing this. And with that, I would really like to thank you for your attention, and yeah, happy to answer questions. My name is Michael Nash, and there's our Twitter handle. Follow us if you're interested. Thanks a lot.